Hello, I'm Manci. I am a second-year PhD student in the Larsen lab. Today, I'll be talking about the application of nanopore adaptive sequencing for COVID-19, specifically a proposed host risk factor in the ACE gene. We're living in the disruption brought upon by the COVID-19 pandemic as you're sitting at home watching my infographic video for Research Day. COVID-19, shortened for coronavirus disease 2019, is a disease caused by coronavirus SARS-CoV-2. COVID-19 is primarily a respiratory disease. Most common symptoms are coughing and fever. In severe cases, however, difficulty breathing, pneumonia, as well as acute respiratory distress syndrome can occur. The highly variable symptoms and outcomes in COVID-19 prompted us to consider the host responses to SARS-CoV-2. Here, we're looking at a genetic polymorphism locus in the ACE gene. Genetic polymorphism is a difference in DNA sequence among individuals, groups, or populations. ACE, which stands for angiotensin-converting enzyme, is an important endocrine component that affects the level of inflammation and blood pressure in the body system. It has been associated with the severity of COVID-19 and is usually in balance with ACE2, the cellular receptor used by SARS-CoV-2 to enter the cell. As shown here, there are 3 genotypes caused by the Lu insertion events for the ACE gene. And these different genotypes influence the levels of ACE protein product encoded by the gene. Specifically, people carrying the II genotype, which stands for insertion on both alleles, has the lowest circulating ACE level, and the opposite is true for individuals with DT genotype. On the other hand, as SARS-CoV-2 enters the cell, it downregulates ACE2. Together with higher levels of ACE in the host, the imbalance likely predisposes individuals to the more severe form of the disease. Interestingly, other risk factors such as aging and high blood pressure are also associated with changes in ACE level regardless of the genotype. Therefore, in addition to confirming the association between the genotype and the outcome of COVID-19 in the US population, we would like to know whether the Lu polymorphism affects the methylation patterns of ACE, and whether there are associations between risk factors of COVID-19 and ACE polymorphism and/or methylation. To answer these questions, we extracted high-quality DNA samples from blood of COVID-19 patients and selected ACE as one of the targets for sequencing enrichment. We are going to use nanopore adaptive sequencing, which is a cutting-edge single-molecule sequencing technology that can simultaneously characterize both DNA sequence and its methylation. The sequencer can make real-time decisions on whether to continue sequencing or reject the molecule based on provided genomic coordinates and real-time data streaming. Therefore, we can selectively enrich for and sequence gene targets of interest, in this case, the ACE gene, from the 20,000-some genes in the human genome. That is what I have for now. Thank you for tuning in. Let me know if you have questions or comments. And happy Research Day!